Most people are familiar with the importance of the microbiome for gut health, but they may not know that the microbiome can affect everything from mood to heart health. Supplementation with probiotics and eating a diet that emphasizes unprocessed plant-based foods can go a long way towards improving the health of the microbiome and your overall health. The microbiome is the community environment and the genetic material of microbes that live in the human body. There are estimated to be at least the same number of microbial cells in your body as human cells. People often associate the microbiome with gut health. However, research has demonstrated the microbiome's role in many areas of human health. Exploration of the microbiome has yielded startling discoveries. The microbes in your body are involved in gut, brain, oral, heart, and immune health, to name just a few. We're only recently discovering the implications of the microbiome on the overall health. Natural interventions such as probiotics and improved diet may help to balance the microbiome and improve many aspects of health. Many factors can contribute to the microbiome's development and disturbances, like a vaginal birth versus a C-section, antibiotic use, dietary patterns, being nutrition and toxin intake, and hormonal cycles, and more. A shift in the microbiome, such as one that lowers beneficial species and or boosts potentially harmful species, is known as dysbiosis. Dysbiosis of the gut has been linked to many chronic diseases and conditions like allergies, cancer, obesity, depression, leaky gut and chronic inflammation, small intestinal bacterial overgrowth, and cardiovascular disease, and more. There's many dietary changes that you can make to help. You can include more fibre, whole grain, legumes, fruit and vegetables, and less animal proteins and saturated fats in your diet. Add omega-3 fatty acids and fermented foods like yogurt, soy sauce and miso. Avoid excess alcohol. Get plenty of sleep to try to manage stress efficiently, and exercise regularly. Some alternative treatments are fecal microbiota transplantation and phage therapy, which involves viruses to attack harmful bacteria while leaving the helpful bacteria and human cells untouched. Probiotics are living microorganisms like bacteria, and they can promote health when they're ingested. Commonly used bacterial species are Lactobacillus and Bifidobacterium genera. Probiotics are generally safe. However, anyone who's very old, very young, or immunocompromised, like cancer patients or patients on immunosuppressive drugs, should consult a doctor before taking probiotics. For digestive health, L. reuteri, Saccharomyces boulardii, and L. rhamnosus may decrease the duration or prevent the onset of diarrhea. Other strains can be effective for relieving bloating, irritable bowel syndrome, reflux, and more. For cardiovascular health, L. casei and L. plantarum may help to improve and prevent the onset of hypertension. L. acidophilus, L. reuteri, and L. plantarum may help to reduce cholesterol levels. For oral health, L. reuteri may reduce plaque formation and improve oral health in people with periodontal disease. Other strains can improve oral health and reduce the occurrence of cavities also. Early clinical research has shown that L. acidophilus and B. bifidum may help to reduce acne. A mix of strains from Lactobacillus and Bifidobacterium species may help to improve and reduce the risk of eczema. Lactobacillus probiotic combinations have been effective in preventing and treating vaginal dysbiosis, and recent findings indicate long-term lactobacillus supplementation may help clear HPV infections also. A specialized dried yeast fermentate from Saccharomyces cerevisiae was shown to reduce seasonal allergies, and L. rhamnosus also with other probiotic strains may reduce the occurrence of upper respiratory tract infections. L. helveticus in combination with B. longum was shown to improve mood and well-being. The human body exists in a dynamic and interdependent relationship with microbes. The skin, digestive tract, airways, vagina, eyes and ears each have a unique microbial ecosystem. There's even evidence that regions of the body long thought to be sterile, such as the placenta, urinary tract, uterus, lungs, blood, and even the brain can also harbour communities of microbes. In healthy humans, these complex ecosystems include a range of microorganisms, from bacteria and viruses to fungi and eukaryotes. Rather than being viewed solely as a reservoir for infection, these communities of the microorganisms are now recognized as essential to our health as individuals and as a species. Historically, the term microbiome has been used to refer to the genetic material of the microbes, when microbiota refers to a community of microbial species. These terms have come to be used interchangeably. The number of microbial cells that we harbour is estimated to be at least equivalent to the number of human cells in the body. The gut microbiome, which is the largest subset of the human microbiome, includes over 1,000 different bacterial species, and it accounts for about 2 to 4 pounds of body weight per person. Furthermore, about 5 million microbial genes have been found in the gut, which is vastly more than the approximate 22,000 protein encoding genes in the entire human genome. Only recently have we begun to understand and appreciate the significance of our evolutionary relationship with our microbial ecosystem. The Human Microbiome Research Project was launched in 2008 by the National Institute of Health as an extension of the Human Genome Project. It's an ongoing worldwide undertaking that seeks to understand microbial communities and their variation in and on healthy humans, beginning with 5 major body regions: the gastrointestinal tract, the skin, the vagina, the respiratory tract, and the oral cavity. A major goal of this project is to create a resource for determining how altered microbial patterns may contribute to diseases and how to restore healthy microbial balance. Thanks to advances in gene sequencing technologies, knowledge about the microbiome is expanding very rapidly. The human microbiome is extremely diverse and surprisingly variable. While only approximately 0.5% of the human genome is variable between individuals, the microbiome can vary dramatically between individuals. Much of this variability is due to the differences in diet, environment, genetics, antibiotic use, and microbial exposure early in life. The diversity and variability of the human microbiome makes its characterization extremely challenging. The largest and most heavily studied components of the human microbiome reside in the gut. Microbiome researchers have estimated that each person is a host to about 160 distinct intestinal bacterial species. Lactobacillus and Bifidobacterium species encountered during birth and are present in breast milk, and these are among the earliest colonizers of the infant gut, and they shape the subsequent colonization with other species. While Lactobacillus and Bifidobacterium species become less abundant in the intestinal microbiome of adults as compared to infants, they have been the focus of much attention due to their beneficial health effects, modulation by the diet and the environment, and possible therapeutic effects when used as a probiotic supplement. Microbes in the intestines produce enzymes that can break down certain food components that human digestive mechanisms alone cannot. A healthy microbiome, therefore, allows us to tolerate otherwise indigestible plant foods and to extract otherwise inaccessible nutrients from them. Microbial enzymes are especially important for breaking down indigestible and partially digestible carbohydrates, including certain starches and fibers. This enhances the body's ability to extract and absorb monosaccharides. These can be turned into energy. Gut bacteria metabolize and activate many plant polyphenols and other phytochemicals, which may have important implications for health. For example, certain intestinal bacteria transform ligands and isoflavones into active compounds which are linked to improve female hormone signaling. The gut microbiome is also equipped with enzymes to digest dietary proteins. Some microbes are more efficient protein digesters than others. Some of the byproducts of microbial protein digestion can have toxic effects, and they may contribute to the negative health outcomes. A Western-style diet, which is high in animal protein and low in indigestible fibers, has a considerable detrimental impact on gut microbiome composition by supporting the species that can best adapt to a high-protein environment. As part of the metabolic process, bacteria secretes a variety of metabolites that can exert beneficial effects on the host. Such metabolites have been termed postbiotics, and they've drawn interest in recent years for their potential to modulate human health and the microbiome. A major metabolic pathway used by intestinal microbes to produce energy is fermentation, which is a metabolic process that does not require oxygen. The main byproducts of microbial fermentation, primarily of digestive fibers, are short-chain fatty acids, including butyrate, acetate, and propionate. These postbiotic short-chain fatty acids are not only a major energy source for cells lining the intestine, but they also regulate immune activity, preventing infections. They reduce inflammation, enhance the absorption of minerals like calcium. They have anti-cancer properties, and they can influence cholesterol levels, appetite, and even weight gain. Byproducts of protein fermentation are somewhat different than those of carbohydrate fermentation, and they include a number of compounds that are potentially toxic. Some of these toxic byproducts are believed to contribute to health problems associated with diets high in animal protein, such as colon cancer and some chronic diseases. Bile acids are the main component of bile, which is secreted from the gallbladder into the small intestine during digestion. Microbial enzymes transform bile acids into a wide array of byproducts, some of which are reabsorbed. In addition to assisting in the digestion and the assimilation of fats and fatty compounds, reabsorbed bile acids and their byproducts help to regulate lipid and glucose metabolism and energy production. How bile acids modulate cardiometabolic conditions such as obesity, diabetes, fatty liver, and heart disease is still being explored. Interestingly, bile acids also contribute to shaping the gut microbiome via antimicrobial effects and interactions with the immune system. Bile acids may be one of the ways to connect diet, which influences the type of bile acids produced, to the composition of the gut microbial populations, which may be supported or suppressed by the presence of certain bile acids. Other byproducts of microbial activity that benefit the human host include B vitamins, vitamin K, and amino acids like tryptophan. Several tryptophan-derived compounds have important impacts on the functions of the immune and the nervous system. For example, approximately 90% of the body's serotonin is made in the gastrointestinal tract from tryptophan. Bacteria that colonize the intestines may also produce other neurotransmitters, such as acetylcholine, norepinephrine, dopamine, and gamma-aminobutyric acid. These neurotransmitters exert a host of peripheral effects, such as the modulation of the stress response, which ultimately influences the brain's function and the mood. Gut microbes play an important role in metabolizing some medications, in some cases activating them prior to absorption. Some of these medications, such as the anti-diabetes drug metformin, appear to rely, at least in part, on altering the gut microbiome to achieve their therapeutic effect. In one case report, targeting the growth of beneficial microbes with a food-based supplement improved both metformin's tolerability and its efficacy. Based on this intriguing case report, researchers carried out a small clinical trial on 10 diabetic individuals. They found supplementation with the microbiome-targeted nutrients agave-inulin, oat beta-glucan, and blueberry polyphenols improved metformin's tolerability and efficacy. Gut microbes also help to process and detoxify harmful environmental compounds such as carcinogens. A critical function of the microbiome is preventing the overgrowth of harmful microbes. Some mechanisms by which it achieves this are by maintaining an acidic pH, secreting antimicrobial substances, inhibiting microbial toxicity, competing for nutrients and resources, preventing harmful organisms from adhering to superficial cells, and activating local immune cells. The microbiome plays an integral role in immune regulation. Almost all tissues and organs, whether in direct contact with, or further away from the microbiota are influenced by the microbiota. Microbes and their products communicate with the immune system. They promote its development and maturation, and they guide immune responsiveness. The immune regulatory role of the microbiome also helps to prevent the dysfunctional immune activity that can lead to allergies, autoimmune disease, and chronic inflammatory conditions. Microbiome disturbances have been linked to chronic inflammatory conditions like periodontal disease, bacterial vaginosis, atopic dermatitis, inflammatory bowel disease, rheumatoid arthritis, and obesity. Furthermore, interactions between the gut microbiome and immune system are important for stimulating the renewal and the repair of cells lining the gut's epithelium, where the gut microbiome resides. The microbiome, particularly in the gut, appears to play a substantial role in regulating metabolism. From the very beginning of life, factors that shape the microbiome have been shown to be indicators of the risk of obesity and metabolic disease. For example, a C-section birth, a formula-based diet, illness, and antibiotic use early in life have all been correlated with higher rates of childhood overweight and obesity. Maternal antibiotic use during pregnancy or breastfeeding has also been shown to impact the infant microbiome and has been correlated with childhood obesity risk. Patterns of gut microbial imbalance have been seen in adults with metabolic diseases such as obesity, non-alcoholic fatty liver disease, and type 2 diabetes. Certain dietary changes, such as consuming more fiber or capsicum from chili peppers, calorie restriction, and weight loss, have been associated with beneficial changes in the gut microbiome, and this may contribute to improvements in metabolic disturbances. Diets high in fat, protein, and sugar, on the other hand, can disrupt microbiome health. The intestinal microbiome undergoes daily shifts in composition that are driven in part by dietary patterns. In an animal model, interruption in the circadian rhythms intended to mimic jet lag or night shift work altered the normal cycling of the microbiome, leading to metabolic disturbances. Emerging evidence further indicates that the relationship between circadian rhythms and the microbiome may be bidirectional, so that imbalances in the microbiome can disrupt circadian signaling, resulting in a cycle-disordered daily biological rhythm and metabolic perturbation. The infant microbiome plays a foundational role in educating the immune system, and it's therefore an important determinant of lifelong health. Its composition is shaped by multiple factors, including the mode of birth, human genetic factors, geography and culture, diet and lifestyle, physiology, illness, surroundings, and contact with other people and pets. In addition, the use of medications like antibiotics and proton pump inhibitors can influence the microbiome. Our first exposure to the microbiome is at birth. During vaginal delivery, the newborn becomes colonized with microorganisms that originate from the maternal birth canal and the skin. Compared with babies born vaginally, the microbiomes of those born by C-section have been found to have more microbes that originate from the mother's skin and fewer microbes that originate from the vaginal canal. Bifidobacterium bacteria, as well as lactobacillus species, are especially important early colonizers of the infant gut, where they help to guide the development of a healthy microbiome and the maturation of the immune system. The mother's vaginal microbiome is critical in establishing the initial gut microbiome in the newborn during birth, and it helps to shape immune system maturation and metabolism. This microbiome transmission from the mother to the newborn occurs during a critical period of the newborn brain's development. Environmental factors, such as maternal stress during pregnancy, can change the vaginal microbiome and its transmission to the infant, and it may affect the neurological development of the infant later in life. Breastfeeding is another opportunity for the transfer of lactobacillus and bifidobacterium species, as well as other bacterial species, to the infant. In addition, certain prebiotic carbohydrates found in human breast milk, called human Mk Oligosaccharides, support the growth of bifidobacterium species, which are important in the infant's gut. where they promote the healthy development and the function of the intestinal mucosal surface and the immune system. Breast milk provides all of the nutrients that are required for a newborn's healthy growth and development. It also contains an array of biologically active compounds that protect and support the newborn during early immune, cognitive, and metabolic development. These are well known to include maternal immune components such as antibodies, immune cells, and cytokines, chemicals needed for immune signaling. Only recently has the importance of specialized breast milk oligosaccharides in establishing a healthy gut microbiome and balanced immune function been appreciated. Human milk oligosaccharides are a complex mixture of small, indigestible carbohydrates that promote the growth of the beneficial gut bacteria, in particular the Bifidobacterium species, abundant in the intestines of healthy breastfed infants, and they're able to ferment these oligosaccharides. By supporting the presence of the beneficial bacteria, human milk oligosaccharides enhance the establishment of normal intestinal barrier function and mucosal immunity. Findings from lab and animal studies also suggest the human milk oligosaccharides may directly impact mucosal and immune maturation and function and exert direct antimicrobial action against infection-causing microorganisms. The differences in the microbiome composition between babies born vaginally versus C-section may explain the increased risk of allergies and asthma, obesity, type 1 diabetes, and neurodevelopmental disorders that are associated with a C-section birth. Introducing C-section born infants to vaginal microbes by swabbing them with maternal vaginal fluids at birth is a new strategy being explored to help to establish a healthy microbiome. Formula feeding also disrupts the healthy infant's microbiome and has been associated with an increased risk of immunologic and metabolic disorders later in life. Even small amounts of supplemental formula can alter the gut microbial population. The infant gut microbiome undergoes changes in the first 2 to 4 years of life, and then it becomes more similar to one found in adults. Changes in the diet and the body contribute to this shift in the microbial profile. Exposure to animals, such as pets, during these younger years is associated with a lower risk of allergies. Microbial patterns can also be altered by antibiotic use, microbes in the surroundings, and illness. After this time of rapid diversification and expansion, the microbiome becomes relatively stable and changes are more gradual. The relationship between aging and the microbiome is still being explored, but evidence suggests the gut microbial profiles of the elderly are different from those of younger adults and are correlated with measures of frailty, poor diet, and health problems. While a healthy microbiome is generally resistant to changes in its environment and it returns to the previous state afterwards, it's nonetheless susceptible to certain disrupting factors. Antibiotics, for example, can have dramatic effects on the microbiome and the ability to recover to the pre-antibiotic microbial balance, and this differs between individuals. Changes in eating habits can also cause profound and rapid alterations in the gut microbiome that can be brief or persistent. Hormonal cycles, travel illness, and aging are other examples of factors that shape the microbiome. A shift in the microbial balance away from a pattern associated with health is known as dysbiosis. A healthy microbiome is highly diverse and characteristically resilient to physiological stresses. On the other hand, a dysbiotic microbiome is generally marked by a smaller number of beneficial species, the presence of more species with disease-causing potential, and lower species diversity. Dysbiosis in the gut increases the risk of immune perturbations both inside and outside of the digestive tract. In addition, gut dysbiosis has been associated with a growing list of chronic diseases and conditions, and these include, but are not limited to, allergies, asthma, autism spectrum disorders, autoimmune diseases, cancer, cardiovascular disease, celiac disease, chronic fatigue syndrome, depression, type 1 and type 2 diabetes, inflammatory bowel disease, irritable bowel syndrome, multiple sclerosis, neurological diseases, obesity, osteoporosis, and cognitive and emotional health. Imbalanced intestinal microflora can trigger chronic mucosal inflammation, and this increases intestinal permeability, and it creates a condition known as leaky gut. The intestinal permeability is controlled by interaction between proteins on the surface of adjacent cells lining the intestinal wall, which form structures known as tight junctions. Zonulin is a protein that modulates these tight junctions, and increased zonulin levels in the circulation are considered to indicate an impaired intestinal barrier. A leaky gut allows the absorption of small food components and bacteria, which can trigger systemic immune activation and inflammation. Gut microbes and microbial products and toxins can also cross a leaky gut. This can lead to immune and inflammatory activation in distant tissues and organs, and is thought to be an important mechanism in the development of autoimmune disease and diseases related to systemic inflammation. While the colon has a complex and diverse microbial community comprising more than 70% of gut microbes, the healthy small intestine has a more limited microbiome. Small intestinal bacterial overgrowth, or SIBO, is a pattern of dysbiosis that's increasingly recognized as a common cause of digestive symptoms such as indigestion, bloating, flatulence, and disordered bowel function, and as a possible underlying feature of irritable bowel syndrome. The most commonly studied antibiotic treatment for patients with small intestinal bacterial overgrowth is rifaximin. This is a non-absorbable antibiotic. In addition, preliminary evidence suggests probiotic therapy, alone or in combination with the rifaximin, may be helpful. Promising results have been reported from trials using probiotics which contain Saccharomyces boulardii or riflexin together with Lactobacillus casei. The fundamental link between the gut microbiome, diet, and physiology has become increasingly apparent, and it underscores the power of diet as a means to maintain a healthy microbiome and thus a healthy body. The gut microbiota is a constant evolution. It varies greatly among individuals, and it modulates metabolism, opening the possibility to predict individual responses to particular foods as part of a personalized medicine intervention. A high-fat diet, especially one including large amounts of saturated fatty acids, has been shown to induce gut dysbiosis, intestinal permeability, and inflammation. A diet high in complex carbohydrates and fiber, on the other hand, supports the growth of microbial populations that are efficient carbohydrate fermenters. These bacteria produce short-chain fatty acids and other fermentation products that support the health of the intestinal lining and have positive effects on the immune and the metabolic functions. A Western-type diet, in particular, high in animal proteins and fat and lower in fiber, selects for bacteria that metabolize proteins efficiently, and those can best tolerate the bile acids secreted in response to the high-fat meal. Microbial metabolism of proteins and certain bile acids creates byproducts that appear to contribute to the development of diseases known to be closely connected to a Western diet, including colon cancer, cardiovascular disease, and metabolic disorders. Conversely, in one study, a Mediterranean-style diet with 35% of calories from fat and a low-fat diet, less than 30% fat, each improved gut microbial communities in subjects with obesity and metabolic dysfunction after 2 years. Prebiotics are carbohydrates that are completely or partially indigestible and that promote the growth of beneficial bacteria populations. These include small carbohydrates called pectin oligosaccharides, large and small fructose-based carbohydrates known as inulins, and resistant starch, legumes, fruit and vegetables, nuts, and whole grains, which are especially high in prebiotic fibers. These foods have long been recognized as the cornerstones of a healthy, high-fiber diet, including the Mediterranean diet, which is known to show benefits regarding a number of metabolic and inflammatory health problems. Although high-fat diets are generally associated with negative changes in the gut microbiome, different types of fat may have different effects. For example, omega-3 polyunsaturated fatty acids, such as EPA and DHA from fish oil, appear to improve the microbial balance and to increase anti-inflammatory immune activity in the gut throughout the body. This may be due to increased positive interactions between the immune system and gut microbes, resulting in higher microbial diversity, increased short-chain fatty acid production, and improved intestinal barrier function. Fermentation, mainly by lactic acid bacteria, has been used to preserve food and to enhance flavor for thousands of years, and fermented foods are recognized for their probiotic and their health-promoting nature. Fermented foods are a source of vitamins, minerals, enzymes, and a range of biologically active, health-promoting peptides, which are produced by microbes during fermentation. Fermented food consumption has been associated with health benefits such as improvements in high blood pressure, high cholesterol levels, insulin resistance, certain digestive disorders, cognitive function, and immune function, and it may lower the risk of cardiovascular disease, diabetes, osteoporosis, and certain cancers. In addition, the living microbes present in fermented foods may act as probiotics when ingested. Abuse and dependence on alcohol beverage has been associated with dysbiosis, leading to inflammation and increased permeability of the intestinal mucosa. Absorption of bacteria, microbial products, and other intestinal content throughout the damaged mucosa may trigger inflammation in the liver and contribute to alcohol-related liver disease. Furthermore, some evidence suggests certain people with alcohol dependence may have increased gut permeability and higher scores for depression, anxiety, and cravings. Changes in the microbiome composition found in this group of people pointed towards the possibility that the microbiome might be a potential target to reduce the risk of relapse in some people with alcohol dependence. Stress and sleep disruption is closely connected and has been correlated with disturbances in the healthy balance of gut microbes. Dramatic reductions in gut microbial abundance and function have been noted following sudden acute stress events, such as a heart attack, trauma, or a brain injury. It's thought that high psychological stress conditions in the body can trigger changes in the gut microbiome, which may lead to changes in the regulation of some neurotransmitters. In addition, due to the bidirectional communication along the gut-brain axis, dysbiosis in turn can trigger dysfunctional activation of the stress response. Stress reduction may be one of the ways to improve the health of the microbiome. Meditation has been shown to reduce activation of the stress pathways and to suppress chronic inflammation, which may support beneficial gut bacterial populations. Furthermore, getting enough sleep and observing normal sleep-wake rhythms may be an important factor for supporting a healthy microbiome. The digestive tract has its own nervous tissue, called the enteric nervous system, that senses and responds to the gut environment, and it regulates gut activity. While the enteric nervous system can function independently, it also communicates closely with the body's central nervous system through a network referred to as the gut-brain axis. The bidirectional nature of this communication explains not only how stress may affect digestion, but also how events in the digestive tract may influence the brain. The gut microbiome is emerging as a key player influencing communication along the gut-brain axis. Gut microbes produce an array of neuroactive chemicals. They help to control intestinal barrier function, and they modulate immune and inflammatory signaling, all of which influence signaling along nerve pathways. Disturbances in the gut microbial community are now thought to be involved in neurological disorders such as Parkinson's disease, Alzheimer's disease, autism spectrum disorder, and in depression, anxiety, and chronic pain. Findings from animal research suggest, most likely via the gut-brain axis, the intestinal microbiome may even shape appetite, feeding behavior, and taste preferences. In a remarkable experiment, researchers transplanted fecal microbes from human Parkinson's disease patients into genetically susceptible mice. This triggered manifestations of the disease, such as impaired movement in the exposed mice, while the same genetically predisposed mice transplanted with fecal microbes from healthy people did not develop these symptoms. Regular moderate intensity exercise is well known to mitigate stress, reduce chronic inflammation, and improve metabolism. Findings from several studies suggest physically active and highly fit individuals have greater gut microbial diversity, with more beneficial microbes than sedentary people. The observed benefits of exercise on immune response, metabolism, and overall health may be mediated in part by its beneficial effects on the gut microbiome. In addition, some researchers have proposed that microbial byproducts may play a role in preserving skeletal muscle structure and function, particularly in the elderly, a crosstalk that's becoming known as the gut-muscle axis. One of the most interesting areas of microbiome research has been fecal microbiota transplantation, which involves the transfer of a fecal suspension containing gut microbes from a healthy subject to an unhealthy subject for a therapeutic purpose. The procedure has generally been performed through an enema or a tube that's introduced into the stomach or the duodenum. However, in a 2017 study on patients with recurring Clostridium difficile infections, an oral capsule of freeze-dried fecal microbes was comparably effective to the delivery via colonoscopy in preventing the recurrences. Fecal microbiota transplantation has become an important treatment option in recurrent intractable C. difficile infections, for which its efficacy is close to 90%. Clinical research, though in its early days, suggests it may also have a value in treating a number of inflammatory and metabolic conditions associated with dysbiosis, such as inflammatory bowel disease, irritable bowel syndrome, obesity and type 2 diabetes, and autism spectrum disorders. Furthermore, fecal microbiota transplantation so far appears to have an excellent safety profile. Bacteriophages or phages are viruses that infect specific bacteria, but they do not infect human cells. Phages are believed to be the most abundant organism on the earth. They're an integral part of the human microbiome, where they're thought to manage bacterial communities by limiting the targeting species. Phage therapy is being explored as an alternative to antibiotic therapy for certain conditions and potentially for eliminating harmful bacteria from the body's microbial communities. An abundant and diverse community of phages inhabits the oral cavity, and specific phages or other enzymes have shown promise for treating periodontal disease and for preventing dental plaque. Phages that target infection-causing gastrointestinal bacteria, such as Vibrio cholerae, which is the cause of cholera, Heliobacter pylori, C. difficile, and some harmful E. coli strains have been identified, and they hold therapeutic potential. Older reports of phage therapy being used successfully to prevent and to treat various other types of bacterial infections include those of the skin, urinary tract, and the eyes, and it's emerged and provides a basis for future research.